Assalamualaikum and good day to all. My name is Shuhaila Mat-Sharani from University Science Malaysia. Today I would like to share my research entitled Conserved and Unique Small Operating Frames in Glaciozyma antarctica. Glaciozyma antarctica is an obligate psychophilic yeast that able to grow as low as a -12 and up to the 20 degrees Celsius. The ability how does this yeast can be grows at the extreme cold temperature could be have a lot of factor, including the small operating frames. These small operating frames, a lot have been missed during the gene prediction because of the small genes. So however, this small operating frame is actually has been found to have essentials in the biological process. So because of that, the aim of this study, we want to identify the conserved and the unique of the small operating frame that have been present in the Glaciozyma antarctica by compared to the other fungi genomes. As you can see in the figure 1 here, we have the genes for the purple color is the ORF, operating frames, and the yellow is the small operating frame, which means that the purple color is more than 80 amino acids, and we can see that the small operating frame is actually less than 80 amino acids. In this study, this is the definition of the small operating frames. So the features is actually same like the genes. We have a start and stop codon. So normally the small operating frame have been found in the upstream region of the genes, and we can found it in the overlap but out frame of the ORF, or the small operating frame can be found between the genes or in the intergenic regions. And lastly, this small operating frame could be found in the downstream region. And also maybe all of this is actually a novel small operating frame. To do this analysis, we do have a combinations of the two approach. So we use the inputs from the Glaciozyma antarctica and 62 fungi genome from NCBI, and then we do the analysis with we have the genome annotations, and we do the genome annotation screening for the first methods, where we filter with the amino acid that less than 80 amino acids. For the second methods, we call it ab initio predictions, which mean we do predict the small operating frame from the raw data, which means we includes the whole genome sequencing, intergenic, introns, and exon. And we do have two software, the GetRF and also SRF Finder. So these two software will predict the small operating frames, and then after that we do the cluster to identify with 100% identity to identify the unique small operating frames. And then we combine both of the results from the first approach and the second approach. We do the clustering to get the small operating frames with the 70% of the identity. And then we do the validations by mapping the Glaciozyma antarctica SRF with the RNA-seq data. And then lastly, we do the SRF characterization. As you can see in the results here, the clusterings for this clustering distribution here with the lengths and also the number of the SRF have been predicted, and the clustering that with the number of the clustering percentage, we have found that 70% we select the 70% similarity, because from this analysis we found that it provides adequate clustering between the species and acceptable length of the conserved region. And also, as you can see in the figure 4, we found that the total of the 150,000 of the SRF in the combination of the Glaciozyma antarctica and 62 fungi genome SRF that has been predicted, we do further cluster into the 6,669 cluster by using the CD hits, and then it includes the 8,322 conserved SRF. And this conserved, which means that because of the SRF that has been homologous or has been present with at least 2 species, which means the presence of the repetitive of the similar gene from the different species is actually as a evidence that those genes actually exist. So we found that the conserve of the small operating frame in Glaciozyma antarctica with the total of the 363. And then we further classified with the SRF characterizations. With that, this results is actually a total of the 352 potential SRF that have been predicted and being validated in the Glaciozyma antarctica, which means it's actually around 4.5% of the small proteins that actually presents in its genomes. So from this analysis, we found that this is 166 is actually from the first approach, genome annotation screening, and then the second one is the gene prediction by using the APBJC approach. We found a total of 1,083 of the SRF, and then we get that 363 is conserved from the previous clustering, and then we do the mapping with the conserved 1,083 total SRF with the RNA6, and we found that 296 of the SRF actually mapped to the RNA6. And with that, the combination and the overlap of these results, we have a total of 352 potential SRF in Glaciozyma antarctica. By further characterize of this potential 352 SRF, we found that 45% have homologs with other genomes with a certain function. For example, here with the small operating frame that has been predicted have function with ribosomal proteins, which means that it involved in the stress response. So this could be helps in the adaptation to the low or the high temperature. And the 159 unique SRF is actually have been found as very unique for the SRF and doesn't have a function, and but it could be important in this Glaciozyma antarctica. And lastly, we do the genes differential expressions of all the 359 of the SRF with the RNA-seq data, and we found that 14 SRF has differently expressed. 6 are upregulated and 8 is downregulated. And from these results, it shows that 2 novel proteins is actually showed a very high expression in the low temperature and could have a potential in Glaciozyma antarctica in adaptation to the cold. With that, as a conclusion, we found that this study is not only expand the understanding of the SRF for the cold tolerance, but we can also demonstrate that by using the homology clustering between the species is actually a useful method to identify the true SRF, and also by integrating the RNA-seq data is also crucial to capturing a large number of the non-conserved and the potentially unique SRF that can be otherwise be overlooked. And last but not least, I would like to acknowledge the grant from the Fundamentals Research Grants FRGS from the Ministry of the Higher Education Malaysia, and we have the second grant from the Ministry of Science, Technology and Innovations, and lastly from the my scholarships, my PhD. And I would also like to thank the APBJC for the travel grants and also for the support to the participants in this APBJC conference. And below here is actually a reference for this study. With that, thank you very much.